For centuries, America told itself a story, a clean story, a story about origins. On one side, you had Jamestown, the rough, desperate, commercially driven settlement planted in the Virginia swamps in 1607. Soldiers, laborers, indentured men, and fortune seekers who came not for God but for gold. Men who buried each other in shallow graves during a winter so brutal it has its own name in the historical record, the Starving Time. On the other side, 13 years later, you had Plymouth, the Pilgrims, the Mayflower, the Compact. The harvest feast folded into mythology so completely that an entire nation eventually built a holiday around it. Pious, purposeful, the righteous founding. For 400 years, these two stories competed quietly in the American imagination. Jamestown was the awkward prologue. Plymouth was the chapter America preferred to begin with, but then the scientists arrived. And when genomic analysis began catching up to bones and the records and the migration trails and the descendants still living today, the story stopped being clean. It stopped being two stories at all. What the DNA revealed was not simply who came first. It was who stayed, who mixed, who vanished into the population, and who, quietly, biologically, became the genetic foundation of what we now call American. The answer, researchers suggest, is more complicated than either founding myth ever allowed, and in some ways more unsettling, and in other ways more extraordinary than anyone expected. This is not a story about which colony won. This is a story about what winning, in genetic terms, actually means, and why the answer may have been hiding in plain sight, in the most unexpected ancestry streams, all along. To understand why the DNA complicates things, you first have to understand what the conventional story assumed. Jamestown, settled in May of 1607 by the Virginia Company of London, was the first permanent English settlement in the Americas. It was not a religious project. It was an investment. The men, and they were almost entirely men in those first years, were sponsored by shareholders expecting a return. They wanted gold. They found swamp, disease, starvation, and conflict with the Powhatan Confederacy. Of the roughly 500 colonists present at Jamestown in the winter of 1609, fewer than 100 survived the spring. The colony nearly collapsed entirely. It survived. It grew. Tobacco transformed it from a desperate outpost into a profitable enterprise. And then something consequential happened. Women arrived. African laborers arrived. First as indentured workers, then catastrophically as enslaved people. And the population of Virginia began to expand in ways no single origin story could cleanly contain. Plymouth, by contrast, arrived with a kind of internal cohesion that Jamestown never had. The 102 passengers aboard the Mayflower in 1620 were not strangers thrown together by a company contract. Many were members of the same separatist congregation, people who had already lived together in exile in Leiden, the Netherlands, before deciding to attempt the Atlantic crossing. They came with community, with shared theology, with the Mayflower Compact, a governing agreement signed before they even set foot on land, a document some historians consider the earliest seed of American democratic self-governance. They also, of course, came with death. Half of the Mayflower passengers did not survive the first winter. The colony at Plymouth only continued because of the intervention and knowledge of the Wampanoag people, a history that is simultaneously foundational and deeply uncomfortable, given what followed. Both colonies grew. Both eventually fed into the expanding British colonial population of North America. And for generations, historians assumed the two streams, the Virginian South and the Puritan North, remained largely separate, culturally and genealogically distinct. Two roots of one tree. What the genetic evidence began to suggest was that the roots were never quite that separate, and that there were other roots entirely, ones the standard story had never adequately accounted for. The first friction appeared not in the DNA lab, but in the demographic record. Historians have long known that colonial-era mortality rates were catastrophic. Jamestown's early death toll is well documented. What is less widely appreciated is how much this reshaped the genetic foundations of early Virginia. When a population crashes and then recovers, it does not recover evenly. Small numbers of survivors or newly arrived groups can contribute disproportionately to all future generations. This is what population geneticists call founder effect, and early colonial America was a study in compounding founder effects, one catastrophe at a time. But the founder effect alone did not explain some of the patterns researchers began finding when they started systematically analyzing the genealogical and genomic data of populations tracing descent to early colonial New England and Virginia. There were discontinuities, unexpected clustering, lineages that pointed not to England South or the Puritan heartland of Lincolnshire and Yorkshire, but to places that shouldn't have been statistically prominent at all. There was also the question of indigenous ancestry. Both colonies made contact, sometimes violently, sometimes through trade, sometimes through intermarriage, with native populations whose genetic signatures were old, deeply rooted in North America, and entirely absent from the origin story England had told itself. Researchers began asking, how much of that contact left a genomic trace? And where did that trace go? The answers, where researchers have managed to find them, have been quietly extraordinary. If this kind of layered human history interests you, consider subscribing, because what follows is where the conventional story really begins to unravel. Genetics entered this conversation, slowly and with appropriate caution. The tools that made this investigation possible are the same ones that transformed ancestry science broadly. Mitochondrial DNA, inherited from mother to child, an unbroken maternal thread stretching back thousands of years, can identify female line ancestry with remarkable specificity. Y chromosome analysis does the same for the paternal line. Autosomal DNA, inherited from all ancestral lines and shuffled with each generation, gives a broader but more complex picture of total ancestry across the whole family tree. Applied to colonial American populations, these tools allow researchers to do something that no document ever could: test the genealogical claims against biology. Not what a family said about its origins, but what the genome actually preserves. And what the genome preserves is not always what the family story says. Studies examining colonial New England ancestry have found that the Mayflower passenger list, carefully preserved, heavily documented, the genealogical pride of millions of Americans, does not always match the DNA, not because the records are wrong, but because the reality of human reproduction in a high mortality, geographically isolated, remarriage-heavy colonial world is messier than a passenger manifest implies. Stepfathers, half-siblings, unrecorded second marriages, adoptions, and the quiet realities of households where not every child shared both parents with every sibling. All of this leaves genomic fingerprints that occasionally diverge from the written record. In Virginia, the picture was more complex still. The complication deepened when researchers began looking at what happened after Jamestown's demographic near-collapse. The colony was restocked repeatedly through the early 1600s. The settlers who arrived in those second and third waves were not randomly selected from the English population. They came from specific regions, specific social classes, specific occupational backgrounds. Many were young men from the west of England, Bristol, Somerset, Devon, not from the areas that fed Puritan New England. Their genetic signatures were subtly but measurably different from the East Anglian and Midlands profiles more common among Plymouth settlers. So, from the very beginning, the two populations were not drawing from the same genetic well. Then came the element the conventional story almost never discusses in genetic terms: the African population of Virginia. By 1650, Africans, arriving first as laborers and then, under the catastrophic legal architecture of chattel slavery, were already a presence in Virginia's demographic structure. By 1700, they constituted a substantial portion of the colony's population, and historical and genomic evidence is clear on one painful and consequential point. The line between enslaved and free in colonial Virginia was not a genetic barrier. Generations of coerced sexual contact, unrecorded relationships, and the brutal power asymmetries of the plantation system meant that African ancestry entered Virginia's white population in ways that were systematically erased from the written record, but not from the genome. Researchers examining colonial Virginia descendants have found African ancestry signals in genealogical lines that the documented record lists as entirely European, not in every case, not uniformly, but with enough regularity to force a reckoning with what the genetic foundations of early American ancestry actually contain. This does not appear in the Plymouth story in the same way. New England's demographic structure was different. The enslaved population there was smaller. The mixing, where it occurred, followed different patterns. What this means, researchers suggest, is that the genomic profile of colonial American ancestry is not a single thing. It is at minimum two things, and probably several more. Three frameworks have emerged from the research to explain what the DNA actually shows. The first is the divergence model. Virginia and Plymouth represent genuinely different founding streams, different English regional origins, different social compositions, different rates of admixture with non-European populations, and those differences persist in the American gene pool in ways that loosely track the North-South cultural divide that would define American history for centuries. The Civil War, in this reading, was not just an ideological conflict. It had distant, diffuse, unintentional genomic roots. The second is a convergence model. Despite initial differences, the two colonial populations mixed extensively as the 17th and 18th centuries progressed. Families moved. Borders were porous. Intermarriage between Virginian and New England lineages was common enough by 1700 that the genomic distinction between the two founding streams had already begun to blur. In this model, there is no Jamestown genome and no Plymouth genome in any meaningful modern sense, only an American colonial genome that absorbed and merged them both. The third framework is the one that makes the conventional story most uncomfortable. It is the hidden ancestry model. It suggests that the most genomically consequential populations in early American history were not the English settlers at all, but the people who were already here and the people who were brought here in chains. The indigenous ancestry that entered colonial lineages through intermarriage, captivity, and quiet integration. The African ancestry that entered through slavery and its brutal intimacies. These streams, largely erased from the official genealogical record, may have contributed more to the actual genetic structure of the American population than the Mayflower Manifest or the Jamestown roster ever acknowledged. The genome, as researchers are finding, does not respect the stories a civilization tells about itself. The most significant body of evidence came when researchers began combining large-scale genealogical databases with modern genetic testing platforms, comparing self-reported colonial ancestry against actual genomic markers in populations numbering in the tens of thousands. The findings were not uniform, but several patterns emerged with enough consistency to be significant. First, Americans claiming descent from both Jamestown and Plymouth settlers showed measurable genetic overlap, but also measurable divergence, consistent with the two populations having distinct, if related, English origins. Second, a notable proportion of Americans claiming exclusively European colonial ancestry carried indigenous North American ancestry markers that did not appear in their documented genealogies. The signal was statistically small in most individuals, but it was present with a frequency that suggested it was not noise. It suggested contact, sustained, multigenerational, and unrecorded. Third, and most striking, the African ancestry signal in American populations with documented colonial Virginian roots appeared at levels that genealogical records consistently underreport. Not dramatically, not in ways that transform a documented history, but enough to suggest that the genetic legacy of enslaved Africans in Virginia is woven into the American ancestry tapestry at a depth that the written record alone would never reveal. What the DNA was showing, in other words, was not that Jamestown one, or that Plymouth one, or that either founding narrative was simply true. What it was showing was that neither founding narrative was complete. Stand in the tidewater marshes of Virginia in early morning, when the James River is silver and still, and the pine woods pressing from every side, and it is possible to feel the weight of everything that was lost and buried here. The bones of Jamestown's dead are still in the ground. The names of most of them are gone. The records are fragments. The stories are silence. Walk north to Plymouth Harbor in late autumn, when the Atlantic is gray and the wind comes off the water with a cold that feels ancestral, and you feel something different, a studied, deliberate memory, maintained and curated, passed down through church records and family Bibles and genealogical societies that have been cataloging descent since the 19th century. Two places, two Americas, both believing they remember where they came from. The genome suggests the memory was always incomplete. Not wrong, exactly, but partial, the way all human memory is partial, preserving what we wish to be true and quietly releasing what complicates it. What the DNA history of early colonial America ultimately reveals is not a winner. It reveals a process. Human history does not move in clean lineages from a single point of origin. It moves in collisions and convergences, in catastrophes that shrink populations to handfuls and then expand them again in unexpected directions, in contacts that leave no trace in a written record but survive, quiet and persistent, in the genome for generations. Jamestown and Plymouth were not two competing origin stories. They were two early chapters in a biological mixing process that began the moment the first European ship made landfall, accelerated through the violence of the slave trade, deepened through indigenous contact, and continued through every generation since. The American genome is not the English genome transported across an ocean. It is something new, layered, complicated, carrying signals from 4 continents in proportions that no founding myth ever anticipated. The Mayflower passengers could not have known they were not the beginning of a pure line. The Jamestown survivors could not have known their descendants would carry ancestry streams that no English record would ever acknowledge. The African men and women brought to Virginia in bondage could not have known that their genetic contribution to the American population would be systematically erased from every genealogical document, and yet survive, biochemically stubborn, in the cells of people who do not know it is there. The genome preserved what history forgot. That is not a metaphor. That is what is actually happening when researchers compare colonial DNA to modern American populations and find signals that the records cannot explain. America did not come from Jamestown. It did not come from Plymouth. It came from the collision of populations that those two places could never have contained, and from the long, complicated, incompletely remembered process of becoming something that had never existed before. The question was never whose blood won. The question, the one the DNA is finally forcing us to ask, is how many people's blood was always in the river, unrecorded, unclaimed, and waiting to be found. If questions like this one leave you want to go deeper into what science is revealing about human ancestry and hidden history, subscribe, because the genome is still being read, and the map is still being redrawn.